Welcome, folks, back to part 2 of chapter 6. So we had just kind of prefaced our discussion that we're about to have, that microorganisms actually exist in community, but it's really useful for us to be able to isolate some of those microorganisms for specific purposes. Maybe they are able to biodegrade a specific plastic and we want to mass produce them. Maybe they're a pathogen and we want to understand them. So it's really essential to be able to isolate individual organisms and then grow them in the lab and do various tests on them. And so we're going to talk about that now. So when we're talking about isolating microorganisms, oftentimes we'll say that we're culturing them in the lab. When we culture microorganisms in the lab, we need to be thoughtful of all the different things that we just talked about. What are their nutrient requirements? What are their environmental requirements? How can we recreate those so that we can actually grow them in the lab? There's a significant number of microorganisms that we haven't been able to successfully recreate the conditions of their growth. We haven't been able to isolate them in the lab. But for the ones that we have, we'll talk about kind of what that looks like. So the first thing that we often want to do is be able to take a mixed environmental sample that will grow on a media, and we'll talk about different types of media in a minute, but essentially think a petri dish, and then isolate a single individual microorganism off of that. And we use a method called streak isolation, where we take an individual colony from a mixed colony. So I'm just going to show you this slide because it has a mixed colony on it, but we'll get back to this method in a minute. So you have a mixed group or community of microorganisms and different types of colonies. And we want to isolate, for example, this really cute yellow one. And so what we would do is we would take one of those colonies, we'd touch our loop to it, which you'll see in the lab, and we'll streak out for isolation, and we will see if we can grow just only that yellow colony. And so here, for example, we're looking for this kind of white colored colony growing on this media here. We can see that we only have that one individual type of colony. So we've probably isolated that specific organism. And so here's another image, what that would look like in this video that's actually from our lab. And so you're looking at E. coli that's been streaked out for isolation. Because they're concentrated on our loop, we first isolate or streak out a concentrated section, then we serially dilute that by then streaking again and again with a clean loop. And I've included a video with the whole procedure for streak isolation in video format below as well. And so you can see this method here for how we would isolate an individual colony off of a petri dish. Now, once we have that microorganism isolated, we need to grow it on different types of media, kind of understand it, for example, so on and so forth. And so oftentimes what we'll do is start off either with a media that's very specific to the microorganism that we want, or is general and tends to be a media microorganisms generally will utilize. Okay, and we'll talk about different types of media in a second. But once we have the microorganism growing, we can actually start to identify some of the characteristics of that microorganism just visually. Okay? And this is through what we call colony morphology. And so you can see here, we're looking at the shape of the colony. Is it circular? Is it irregular? The margins of the colony, so how those margins are they very smooth, or do they tend to kind of have ridges, so on and so forth? The elevation of the colony, the size of the colony, the color, and the transparency of the colony. And that will start to give us some information about what that organism is and who that organism is. Okay? But we tend to need to get other information as well. And so let's talk a little bit about the different types of media that we might use either to grow, identify, or manipulate a microorganism. Okay, so we have these 6 types of media: defined media, complex media, selective media, differential media, anaerobic media, and transport media. And some of these overlap. So defined media would be, actually, it's not very common that we use this in our lab, but it would be if you were trying to grow a microorganism with very specific nutrient requirements. And you would have a very specific ingredient list of chemicals, like for this, for example, for culturing E. coli, although E. coli will grow in other types of media as well. And it has the very specific nutrient requirements of that very specific microorganism. And so that microorganism is the one that tends to grow in that media, selective of other microorganisms. Okay. Now, like I said, we don't use very much, in this class, we actually don't use any defined media, but it is important in some situations where you're working with a very specific microorganism. What we use most often in our labs is what's called complex media. And complex media is where you take something like a broth, like a beef broth or a vegetable broth or soy broth, that already has a complexity of nutrients in it, but that complexity is not exactly specifically defined, right? It's not like you went and, measured out a specific type of nitrogen, a specific type of carbon in very specific amounts, so that when you took that beef broth or you took that soy broth, you know that there's a complexity of nutrients in there that generally encourages growth of microorganisms, okay? And so the type of media that we use most often that is a complex media in our lab is trypticase soy agar. Excuse me. And this is a media made off of soy, and you can use it in a liquid form like this broth or in various types of solid forms. In fact, basically any media can be poured in different kind of forms, either as a plate so you can easily see colonies that are growing, or as a broth so you can encourage a lot of growth, or as a deep. You'll see how we use deeps when we're looking for growth in response to various levels of oxygen, or a slant, which tend to be for long-term storage. Okay? And so for this type of media, many types of organisms will grow. Not all organisms, microorganisms, but many types of microorganisms will grow. So if you just leave this open in the lab, you'll get various types of yeast and fungi and bacteria growing on there happily, right? You do need to be thoughtful of oxygen requirements. So if you leave it open in the lab, you'll grow only aerobic microorganisms, whereas if you put it in a jar and remove the oxygen, which I'll show you some examples of that today, then you'll grow only anaerobic microorganisms. But it's a kind of a general type of, food. Think of, in the macro world, a food that lots of people like pizza, for example. Not everyone likes pizza, but generally speaking, if you have a pizza party, lots of people will be happy and they'll be able to eat it and enjoy it, right? And so these TSA plates, also we'll use LB plates and LB agar. These are these complex media, and they're just general kind of foods that microorganisms will be able to grow on, okay? So they're kind of the pizza of the lab world, the microbial lab world, okay? And so you could then start to layer in complexity, okay? Instead of just having a TSA or an LB, which is just a general complex media, you might start to add things into the media to make it a little bit more complex and to select out certain organisms or to differentiate between certain organisms that are in that environment, right? And so, for example, you might change the pH of that TSA, that trypticase soy agar that I had showed you before. So instead of just growing lots of different microorganisms that are in the environment, it only grows those that can live within a more acidic environment. For example, lots of fungal colonies, okay? Or you make it more basic and it only grows organisms that can respond to a more alkaline environment. And so what you've done is taken a complex media and made it what we call selective. Selective meaning that instead of growing all of the possible microorganisms, it selects for just microorganisms with a specific characteristic. And we're going to use a lot of different types of selective media. For example, mannitol salt agar, which we'll talk more specifically about in a minute, selects for microorganisms that tend to have specific characteristics. And in the clinical setting, these characteristics tend to align with Staphylococcus aureus infections. And so MSA plates can be used to select out for organisms that might possibly be Staph aureus. Okay? And so by manipulating the components of the media, we can make them selective. So I gave you the example of changing the pH. Mannitol salt agar changes the salt content, so it has a higher salt content, and the sugar content. So there's a sugar called mannitol that's added into the media that's specifically utilized by some microorganisms and not others. And so when we change the component of the media, we've changed what we'll actually see grow on the media, and we've made it selective. Okay? So let me give you some examples of selective media, and then we'll layer on a little bit more complexity in a minute and talk about some other things that we can see with media. Now, another example would be phenylethyl alcohol agar, where we essentially have some chemical components, including alcohol, that select out for organisms that are gram-negative and only grow gram-positive organisms. So if we plate multiple different organisms, the ones that will grow will be gram-positive organisms and not gram-negatives. Just to note, the scientific formatting of these names is incorrect. So we want to make sure that we're always formatting our scientific names correctly. Okay, so these are examples of media that we have, a complex nutrients that are in there. We have some sort of change in the media, either pH or, for example, the adding of chemicals, which makes them selective. Selective meaning that we see some organisms grow and other organisms are inhibited from growing. Okay. We could also have media that is what we call differential. Differential means that we can look at the plate and based on characteristics in the chemical reactions of the plate or in the colonies themselves, we can see specific characteristics of those organisms, okay? So here we have TSA where we just grow a bunch of different organisms, but this is an example of blood agar plates where we can actually see differences in the way that organisms interact with blood just visually by looking at this plate. And that means it's differential, it's differentiating between the different types of organisms. Now, blood agar is a complex media, so it grows many types of organisms, but includes blood. And when we include the blood, we can see some organisms that are not pathogens, they don't interact with blood, just growing. So for example, E. coli would be what we call gamma hemolysis or no hemolysis. Hemolysis referring to the lysis of the blood cells. So if we do have lysis of the blood cells, then we might get beta hemolysis. And that might say, oh, there's some sort of streptococcus, for example, species that's in the sample, okay? Or you might have other types of streptococci, like Streptococcus pneumoniae, that only have partial lysis of blood cells. And we can see that as alpha hemolysis. So out of the different microorganisms that we've grown on this plate, we can see very clearly how they interact with blood cells because of the way that we've constructed this media, okay? And so we're differentiating, because this is a differential media, between the different types of organisms, okay? Another example of differential media are the phenol red carbohydrate broths that we're going to use in our lab. And so it's very indicative of a microorganism, what sugars they tend to use in their metabolism. So we can have complex media that grows many types of microorganisms, but has specific sugars in there, okay? And those specific sugars will be fermented. If they're fermented, we get a color change. And if they're not fermented, we don't get a color change, right? And we can also see whether gases are produced. And so if we see a yellow color change and or gas being produced, then we know that that specific microorganism is utilizing that specific sugar. And so that allows us to differentiate between different microorganisms that use different types of sugars in their fermentation. Another differential media that we'll use in the lab is gelatin agar or gelatin media, where we actually use a slightly modified test that's in a tube, but the idea is similar, is that you can have organisms grow on this media because it does have complex nutrients in it, but the ones that utilize gelatin, because we add gelatin to the media, will actually have a halo around them because they've actually digested or hydrolyzed that gelatin. In our case, we'll see liquefaction of the media because of the gelatin used, right? And so you have many organisms that might grow, but you can differentiate between those organisms based on their gelatin hydrolysis. Same for starch. We'll use a plate that has starch added to it. If the organism utilizes the starch, then it will, be hydrolyzed and it won't be present in the media after incubation. And we'll see when we add iodine, we'll see a halo around those organisms, whereas organisms that don't utilize starch, they'll just grow, but they won't show this halo when we add iodine. And therefore, we can differentiate between organisms that do have the enzymes to break down starch, like amylase, and this one right here, that do have the enzymes to break down amylase and that don't have the enzymes to break down. That do have the enzymes to break down starch, for example, amylase, and that don't have the enzymes to break down starch, like amylase. Okay. So another example of a differential media that we will use in the lab is thioglycolate media. This is a broth that has a number of chemicals in it, and we'll talk about them in the lab. But essentially, it has a chemical that indicates the presence of oxygen, and this is called resosirine. It also has some oxygen scrubbers like thioglycolate that actually remove oxygen from the media. And so at the very top of the media, we'll inevitably have oxygen because this is an open cap or a cap with a gap in it. So oxygen does come into the tube. And any organisms that grow at the very top of that are going to be our strict aerobes, as we had mentioned before. Whereas organisms that grow at the very bottom, where the thioglycolate has scrubbed the oxygen out of the media, are going to be our strict anaerobes. And then we have some, variation in between. And so this thioglycolate media is growing many different types of organisms, but we're able to differentiate them based on their oxygen use. Okay. And so this is kind of, when we're talking about media in the lab, we're going to ask the question, is it differential? Does it differentiate between the organisms that grow on it or not? Are we just kind of seeing generally organisms growing? Okay. And so we've talked about selective media and differential media. Oftentimes you'll have selective media that is also differential. Okay. And so the example that we had used before of mannitol salt agar is actually selective and differential. So it selects out for certain organisms, and it differentiates between certain organisms as well. Okay. And so I'm going to show you some examples. Oh, here, let's start with mannitol salt. So we said that Staphylococcus aureus, or we had briefly mentioned, but let's talk about it more. Staphylococcus aureus is an organism that is a halophile. It likes really salty environments because it grows on our skin, right? So there's different environments on our body, just like there's different environments on the in various ecosystems. And our skin tends to be dry, and it tends to be saline because our glands are producing sweat and other salty components. And so on our skin, you have a lot of salt. So these are halophiles. They also are characteristically ferment mannitol, a sugar called mannitol. And so there's a test that seeks to be able to identify organisms that have these characteristics. Specifically in the medical setting, these are used to find Staphylococcus aureus. Now, there are other organisms with this characteristic that mimic the growth of Staphylococcus aureus. Those are the ones that we're going to use in our lab. So you will get to see these results as well. So what happens here is the mannitol salt agar, the salt actually selects out for a number of different organisms. So specifically gram-negative organisms, they have just less protection around their physical cell. And so they tend not to be able to exist within these high salt levels. And then generally, organisms that can tolerate salt, they'll be there, they'll be present. So it is selective for halophiles or halotolerant organisms, and generally speaking, for gram-positive organisms, because the gram-negatives can't tolerate that much salt. And then it is also differential. So you might have gram-positive organisms that are halophiles, but they don't ferment mannitol. And so they'll just grow just kind of on the media without changing its color, without interacting with the mannitol. Whereas organisms that do ferment mannitol, so there's sugar mannitol is in the media, then we'll get a pH change, and then there's an indicator that turns yellow when we get that pH change, and we'll know that yes, this is a halophile that is gram-positive and also ferments mannitol, and it will be distinguished from halophiles that are gram-positive but don't ferment mannitol. And so in that way, it's both selective and differential. Okay. We also have McConkey agar. McConkey agar allows us to look for gram-negative organisms. There's a different set of chemicals that select out gram-positives and tend to allow gram-negatives to grow. And in this case, we are looking for the differential growth based on the fermentation of another sugar, a different sugar. And so what you're going to see is if you have that sugar being fermented, so lactose is the sugar, then you're going to see pink colonies or a pink halo. And if that sugar or lactose is not fermented, then you're not going to see that pink color. So either it'll just stay red or it'll be yellow, okay? And so in that way, we're selecting for organisms that are gram-negative. They're the only ones that will grow. We'll select out gram-positives. And we're looking for the differential characteristic of different organisms, whether or not they can ferment lactose, in this case, E. coli can, or not ferment lactose, in this case, Salmonella can't, okay? And so in that way, in the lab, almost every day for a series of labs near the end, you're going to see this table here. What is the media? Is it selective? Is it differential, right? And so we're going to be working through those. And that gives you an idea of how we take media and manipulate it so that we can either identify microorganisms or learn more about them in the lab, okay? Now, I promised that I would show you how you could do any of these tests with aerobes or anaerobes. Save this thioglycolate broth, which has kind of embedded within it a test for oxygen, response to oxygen levels. But you could do any of these tests other than that thioglycolate one on either aerobic or anaerobic cultures. And the way you would do that is to put it in a jar, and we have bags that you can actually chemically, that you open up and they will chemically scrub out the oxygen from that environment. There's also a less expensive way of doing this and just do a jar, put a candle in the jar, the candle will burn until all the oxygen has run out, and then that will be an anaerobic jar. And so I have a video of that in the description below. Either of those methods will allow you to incubate these plates in either an aerobic or an anaerobic setting. Okay. So the next thing that we want to talk about is microbial growth and growth phases. So go ahead and pause this video, finish up your notes and your diagrams from this part of the lecture, and then jump into the next part of the chapter 6 lecture series. Okay, see you there.